Her research focused on understanding biological processes through knowledge of structure of key protein molecules. She has a major contribution to understanding of catalysis and regulation in glutamine aminotransferases and phosphoribosyltransferases transferases by solving and interpreting crystal structure of several enzymes of each type. She has also solved crystal structures of photosynthetic proteins, leading to a new understanding of their function. She has also contributed to development of methods for rapid determination of protein crystal structures, particularly using synchrotron X-ray sources. She is native in Pennsylvania. Janet studied chemistry as a National Merit Scholar at Indiana University of Pennsylvania and received her Bachelor of Science in 1973. Then she did a PhD in 1978 at the University of Wisconsin-Madison. The postdoctoral studies she did in Naval Research Laboratory with Wayne Hendrickson. 10 years later, she established a program in structural biology, Purdue, where she remained as a professor of biological sciences, until moving to the Life Science Institute, where she's a research professor, and also she is a Margaret Hunter Collegiate Professor of Life Sciences at the Department of Biological Chemistry, University of Michigan Medical School. She has been a visiting scientist at the European Molecular Biology Laboratory and the European Synchrotron Radiation Facility in Grenoble, France, and lectured at numerous international schools on structural biology and synchrotron radiation. Also was a part of many SAC facilities, at synchrotron facilities. In addition to, she is also GAC CAT director at APS and a member of Commission on Synchrotron and XVL radiation since 2011. So today's presentation is entitled Virulence Factors and Host Defenses to Viral Infection. Janet, please come up.
Thank you, Pavel, for that lovely introduction, and thanks to the program committee for inviting me to speak here to you today. And also, I would like to thank the crystallographers from India who I've met, especially the students who've been extremely hospitable. This is turning out to be a great experience. So in the talk today, I would like to tell two stories. The first story is a published one about the virulence factor known as NS1 from several flaviviruses. And then the second story is another one it's another virus story, but it's a rather different one. And this is about the restriction factor for HIV known as ApoBec3 or A3H. And that's an unpublished story. So here we go. I don't have to tell people in this country, excuse me, about the challenge of dealing with flavivirus infections. These viruses include some real public health concerns, including West Nile virus, dengue virus, which is endemic in all tropical regions of the world, yellow fever virus, Japanese encephalitis virus, and more recently, as has come to our attention, the Zika virus. These are even though the diseases they cause are quite different from one another, at the molecular level, these are highly related viruses. They're in the same genus, the flavivirus. Yellow fever virus is the type member, and there's a very effective vaccine for yellow fever, but the world stock is not very large anymore, and we narrowly missed an epidemic earlier this year. It was a small outbreak in Africa, and thanks to the generosity of many nations across the world in donating large fractions of their yellow fever vaccine stock, we managed to avoid an epidemic. But there are no vaccines, no effective vaccines or treatments, antiviral treatments for the other flaviviruses. So a little bit about the biology. The flaviviruses are a membrane-enveloped virus. Here's a picture of the virus particle. This is from a paper published a year ago. This is the Zika virus from Michael Rossman and Richard Kuhn. It's a host-derived membrane. The viral genome is a positive sense RNA, single-strand, and it's 10.5 kb in length, and it encodes one single polyprotein of about 3,400 amino acids. And what's shown on this slide is a cartoon of the open reading frames. Most of the genome is coding. The open reading frames for several proteins that are generally divided into structural, i.e. the proteins are part of this mature virus particle, or non-structural, they are not part of the mature virus particle. So we're concerned here with the one called NS1. So that polyprotein, well, first of all, it's a plus sense RNA virus. So when it gets, the genome is released into a cell, it stays in the cytoplasm, it looks like messenger RNA, and it's immediately translated by the host ribosome into this large polyprotein, which is inserted in the endoplasmic reticulum membrane through a series of translocate and stop translocate sequences in the polyprotein. And then it's chopped into its mature protein parts by the viral protease for cleavages that occur inside the cytoplasm, and by host proteases for cleavages that occur in the ER lumen. And at the time we started this project, there were 3D structures of all of the outside the membrane bits of the polyprotein, with a single exception of the NS1 protein. And NS1 is a little bit mysterious. It has many functions. The one that's really essential to the virus is that it's very important, even though it's on the outside the membrane, out in the ER lumen side of the membrane, it's essential for replication of the viral genome, acting on the opposite side of the membrane from where all the replication machinery is. So in this picture, can this pointer, that's much better. Now I can see. NS1 is here, if you look at the right slide. And all of the business happens on, this is continuous with the cytoplasm. So here's where the RNA replication takes place and new virus particles are made. But then NS1 has another life. It's known when it's inside the cell like this to be a dimer. Eventually it gets secreted through the secretory system and ends up in the circulation of infected patients as apparently a hexamer. And a few years ago, there was a very low resolution EM reconstruction of the secreted hexamer from Felix Ray and Marie Flamont. And they also ripped this thing apart and discovered that it contains not only protein but also lipid molecules. The NS1 protein had eluded anybody's attempt to purify it. It's highly conserved, 352 amino acids. As a secreted protein, you won't be surprised to hear that it is replete with disulfides. It has 6 disulfides, 12 cysteines, and it is also glycosylated. The sequence identity is in the range of 40 to 80% among different flavivirus NS1s, and it tends to be membrane associated. So the first thing we did was figure out how to make the protein, and this really was the work of the gifted and patient and persistent Clay Brown, who is a biochemist and cell biologist. And he, at the time we started, we had 4 cDNAs for 4 different NS1s from different viruses. He cloned them into a variety of vectors that had 3 different secretion signals, 3 different purification and solubility tags, and he put them in 2 different insect cell lines, which is where we decided to make the protein. And this came up to be 72 expression tests, and each one of those was tested for production of the protein inside the cell as well as secreted into the media. Now we know the mosquitoes, so flaviviruses are carried by either mosquitoes or ticks. These ones all by mosquitoes, and not much is secreted into a mosquito vector. It's mainly in us that the NS1 is secreted. And sure enough, Clay found it inside cells. And what we noticed was that what he discovered that was really key to getting this protein made was that we needed a little bit of detergent to persuade it to leave the membrane. If you broke the cells, it would always come with a membrane fraction. And then did the first purification step in the presence of that amount of detergent and then got rid of it after that, or at least carried on the purification without it. But as we did successive gel filtration columns without detergent, the protein went from looking like a hexamer, looking like a dimer, running like a dimer and looking like a dimer in negative stain EM, to beginning to aggregate as hexamers, apparently. But for the most successful protein in the initial round of work, which was from West Nile virus, both of these fractions crystallized equally well and in the same crystal form. Crystals grown by Jamie Konworski and at the time Tom Jerkue. And so the NS1 protein we quickly discovered never met a membrane that it didn't love. And here's an example of, again, with EM, the purified NS1 protein, some liposomes that Clay made, and if you mix these together, you bust up the liposomes. It really, really likes membranes. And this is very consistent with the property that we think NS1 is membrane associated opposite the replication machinery. So the crystallography for this project was done by David Akey, and he decided to try and do sulfur SAD, single wavelength anomalous diffraction at an energy that is well away from the sulfur edge. But this was right after Chunlu and Wayne Hendrickson's paper in Science about the success of sulfur SAD with several structures that they had previously solved by other means. In this case, the sulfur anomalous signal we estimated was about 1.5% of F. And so Jamie grew crystals, David screened them, and we had space group P321 or some variant thereof, and diffraction typically to 3.2 at this energy, and David collected many data sets. The statistics were looking reasonable at low resolution, not very reasonable at high resolution. However, if he combined, this is for an 18 crystal data set, if he merged the data, this is anomalous correlation coefficient and anomalous signal, the merged data from 18 crystals was far superior to any individual data set. So he worked with an 18 crystal data set and then used ShellX to try and find the anomalous scattering centers. Because of the disulfides, you get a little square root of 2 boost in the anomalous signal, and it wasn't clear how many were in the asymmetric unit. Could be 4, unlikely, could be 3, more likely, could even be 2. And so we didn't know how many sites we were looking for, but it became apparent after a lot of experimenting with ShellX that we were seeing something that looked like twofold non-crystallographic symmetry in the apparent sites that we were finding, and i.e. very high solvent content. So this was done with the data to 5.2 angstrom resolution where there appeared to be an anomalous signal. He calculated initial phases in that data set to 4.5 angstrom in ShellXE, did some phase refinement, real space solvent flattening, and I maybe also averaging, I'm not sure. Anyway, and then shifted to a better data set that we had from a 12 kilovolt experiment and 1 crystal, and worked with in DM and stepped out very slowly. It's 2 cycles of phase extension, a la the old virus crystallography method of coming up with an interpretable map, out from 4.5 to 3 angstroms, and obtained a really beautiful and highly interpretable electron density map. So because we didn't know anything about the sequence of NS1 does not look like anything else that's in the structure database. So we had no idea what the fold would be, but we fully expected it to look like something we'd seen before. But we needed to have a map that we could build a new structure into directly. And we got one. He got one, and that was it was a big day in our lab. And then before I go on, I'll say that then that was a couple years ago. That paper was published in 2014. Last year, when everyone was going whole hog on Zika virus, we also solved a Zika virus structure and got crystals that diffracted really well from that NS1, and that's summarized here. And now I should point out there are several structures. So in the initial publication, we reported, this is the initial West Nile virus one that I will tell you a little bit more about in a minute. In the dimer form, we also published at the same time 2 hexamer forms that didn't diffract nearly as well, and then the Zika one at higher resolution. And these are full-length NS1 proteins. And in the meantime, several labs had discovered how to make the C-terminal half of the protein, published that, and then there was a lower resolution structure of the full-length Zika virus one from George Gao's lab last summer. Okay, so that's what we know about structure. So now I'm going to tell a little bit about the structure. It's really a dimer. It's inherently a dimer. And if you know anything about protein structure, you'd never believe it could ever be a monomer. But I will talk about this structure, and I will come back to the hexamer, and the hexamer is made of 3 dimers. And there are landmarks on the structure, and I will try to point them out as I go along. And these are perpendicular views of the dimer. We saw the entire polypeptide from, in here, one polypeptide is a rainbow and the other one is gray, from the N-terminus right there to the C-terminus right there. And there's a 20 amino acid loop in each monomer that was disordered. And this is the face-on view. It's a very flat structure, and this is the end-on view, viewed from the top end. Okay. It did not look like anything we'd ever seen, so we had to make up names. It has 3 clear structural domains, what we called the beta roll, a little guy at the amino terminus, what we called a wing, following, and then the beta ladder, this is the entire C-terminal half of the polypeptide. And that's what other labs have published structures of, looks exactly the same. I want to start there because this is really the backbone of the protein. This is a new fold. Each monomer contributes 14 beta strands, and it's a continuous anti-parallel sheet, back and forth, back and forth, back and forth. And you can see it looks like a ladder, a little bit curved. And then there's a long loop, about 50 amino acids, no secondary structure whatsoever, but extremely well ordered. There are 50-some hydrogen bonds between main chain and side chain atoms that hold that structure and fix it to the backside of the ladder. The wing domain, which is called the wing because it sticks out the side, one in each monomer, is the only thing with helices in it, and it's also the only part of the structure that has any resemblance to anything that we've seen before, and it most looks like a superfamily 2 helicase domain in 2 antiviral proteins, proteins of the innate immune system that detect double-stranded RNA in cells and mark them for destruction, RIG-I and MDA5. Maybe viral mimicry of that, probably not. This doesn't bind RNA and has no special positive charge. It may be just a coincidence, who knows? It's early days. Well, I should mention that when NS1 is secreted, it travels around in the serum of patients, especially people who have dengue virus infections, And the higher the worse the infection, the sicker they are, the higher the levels of NS1 in the serum. The highest that's ever been recorded is 50 micrograms per milliliter, which is a huge amount of protein. And we don't know what it's doing out there, but it's interacting with certainly things in the immune system, not this protein, which is not MDA5 or RIG-I, which are inside cells. Okay, the beta-role, super dimeric. And you can see it's a little roll. There are hydrophobic side chains in there holding it together. And you can see how it's not exactly intertwined, but almost. And it's the site of a couple amino acids that have been known to have something to do with the replication function. And that's pretty interesting because if we look at the protein end on, so now this is our, where's my pointer? There it is. Looking at the latter end on, the wings on the side, the beta roll protrudes on one side, and this, if you think about the structure this way, it's like being in a city where you walk 2 blocks and you're in a completely different neighborhood. So this neighborhood out here has a flexible loop, it's got the glycosylation sites, it has that spaghetti loop, and it's very polar. This neighborhood in here is hugely hydrophobic. So here's a electrostatic surface potential drawing. Here's this picture from end on again, down here. And the green stuff that you see down here is this electron density, and this is apparently leftover detergent from the purification. The Triton is notoriously sticky. And at the threefold axis of that crystal, 3 of them come together, and of course, this greasy bit's on the inside, and I remind you of its ability to bust up liposomes and the fact that it's apparently bound at the membrane during its replication function. So this is a nice way that the structure sort of goes together with a lot of other data about this protein. And then I need to tell you something about the hexamer. So in the hexamer, remember I said this was a particle that Felix Ray discovered had both lipid and protein in it. And so you probably won't be surprised to hear that the hydrophobic bits of NS1 are pointing to the inside of that hexamer particle when you take 3 dimers, one shown in ribbed form and 2 as solid surface, and put them together. And we had, and of course there's no lipid in here. This is a lipid particle, proteolipid particle. The reprojections of the crystal structure of the dengue in that hexamer form, dengue NS1, match negative stain images that we got of the purified protein. So to our surprise, now I'm still talking about the hydrophobic surface, but to our surprise, when we solved that high resolution structure of the Zika virus protein, suddenly there was electron density for those missing 20 amino acids. That was cool, okay. But more interesting was the fact that they didn't go where we thought they were going to go. So now I've gone and changed colors on you. Everybody's gray. There's the beta roll in blue, some greasy residues pointing down, and here's where that missing 20 is. Well, the missing 20 don't do this, the missing 20 do that. They point down in the same direction as the hydrophobic surface, and they expand the hydrophobic surface. So there are some conserved residues in here. Here's the structure of it. These aromatics are standouts there, and those are those yellow marked residues in here, and they are conserved. And now I want to show you a little movie. This is the Zika virus protein with that ordered loop crystallized as a dimer, not as a hexamer. But if we take the other hexamer structures and put the model build the Zika onto it, the structures from the 3 viruses that we've solved are virtually identical. You can see that that green new loop out in the wing domain, the yellow is the wing domain, is what's making all the protein-protein contacts in this hexamer particle. And I like to show this view of it because it really points out how open that structure is, and there's plenty of space for, and the greasy beta roll is pointing inside, along with the orange protrusion, which I will get to in a minute, and it's just waiting for some greasy molecules. Okay. So one more little piece of data. The residues 10 and 11, which are on a loop in the beta roll, had been discovered to be important for replication. So Michael Diamond at Washington University in St. Louis swapped an asparagine lysine for an arginine glutamine from dengue West Nile, put the dengue ones into West Nile, and suddenly found that it was really sick. This was not a very healthy virus. He passaged it for many times, and it popped back into healthiness, and he sequenced what popped back into healthiness, and lo and behold, the change to the sequence occurred in this little protein NS4B over here, and then he went and got some so-so data to show a physical contact between those proteins. So this was sort of another satisfying result that, oh, this part is involved in somehow in replication. We don't know exactly how. So we went and looked at this orange piece, this piece that hangs down here that's part of a connector between the wing and the beta roll, and it has a fairly conserved sequence, and made some radical substitutions to aspartate that were completely dead, phenylalanine to alanine, and then a double mutation, glycine to alanine and phi to alanine, which was dead. And we had a sick virus with, what you would think would be a pretty modest substitution. This is an exposed phenylalanine, but it's not. You can grow the virus. You can grow the virus in cells, but you have 3 orders of magnitude less RNA produced in cells and also as infectious particles. So these greasy bits are very important to the virus life cycle in ways that we don't have a molecular mechanism for. So then the last bit of data that I'll tell you about for this project is that we then did a similar experiment to the one Diamond had done, where we said, what is the same in the 4 serotypes of dengue virus that are in circulation? What residues are conserved there, but they're different in the West Nile. So for proteins that are 40 to 50% identical in sequence like these are, usually single amino acid substitutions from one to the other don't really make much difference. So we did a whole bunch of these in collaboration with a collaborator virologist, Richard Kuhn, at Purdue, and picked up 2 new phenotypes where these substitutions, not together, separately, generate a new phenotype where NS1 has reduced secretion outside of cells and reduced release of infectious particles. And then the second phenotype, which came from 2 other different substitutions, was that the particles that made virus particles, but the particles were not as infectious. How does this work? No idea. But it's a hook to go and find what else the NS1 is doing. And we, of course, are not the only people who have jumped on the structures and tried to understand the virus molecular function better. And here are some papers that have appeared since we wrote the initial, published the initial structures. And I do want to point out 2 here, one from Ava Harris' lab at Berkeley and one from Paul Young's lab in Queensland in the same issue of Science Translational Medicine. And they discovered that if you take the NS1 protein and put it on cells or even in an animal, you can generate, in absence of the virus, you can generate the vascular leak phenotype, which is the hallmark of the dengue hemorrhagic disease. So this protein has some pretty important functions for the virus, and we're just beginning to get a handle on them. And I think yes. So this is my little summary about the NS1 story. What is going on with this protein? It's obviously multifunctional, and my view of it from all that we've learned is that this infected cells have a lot of NS1. If you think about what it takes to make a virus particle, one particle takes one copy of the RNA genome. It needs 180 copies of the capsid protein, which binds the RNA inside the virus particle, this little membrane protein, and the envelope protein, which is really what makes up the outside. 180 copies of each of these. You certainly don't need 180 copies of the enzymes, which are not part of the virus particle. And nor would one guess do you need 180 copies of NS1. But as far as we know, when the polyprotein is produced in infected cells, unless it happens to fall off, the ribosome zips along and makes the whole thing, which seems awfully wasteful. But the very first protein after the structural ones is NS1, and there is a lot of it. And a lot of it gets secreted, as we've seen. And it may be helpful in remodeling the endoplasmic reticulum membrane. This is a tomogram from Ralph Bartenschlager, and which they colorized to show this pointer, to show the membrane network, and then the black regions, which are the ER lumen, and to see how that membrane is remodeled during a virus infection. And I think NS1 may have some roles in this, somewhere in this process. And then who knows what it's doing when it gets outside the cell. So the acknowledgments for this part of my talk are that at the University of Michigan, David Akey and Clay Brown were the prime movers in this project, along with Jamie and Tom, helping out. And then we have collaborations with the Kuhn Lab at Purdue and one postdoc and 2 students in his lab. Yorgos Skiniotis, who was at Michigan, now Stanford. Samnat Dutta, who was at Michigan, now in Bangalore. And Jeff Tarash to help with the EM. Ted Pearson is a virologist at NIH, who's also collaborated on this. And of course, all of our photons come from GMCA at the APS, and Craig Ogata helped with the sulfur SAT experiments in the beginning. We've had funding from NIH and from the Martha Ludwig Professorship, which I was very proud to have for several years at Michigan, and the Perigo Foundation supported one of the students. Okay, so that's the end of part 1. That was fast. So now I'm going to switch and tell you a completely different story. And this one is not published. It's this close, but it's not published yet. And this is work that was done in my lab by a very talented graduate student, PhD student, Jennifer Bone. And this is about a protein called ApoBec3H. And this has to do not with the flavivirus, but with another positive sense RNA virus with HIV. Oh, actually, before I leave the flaviviruses, so dengue is endemic in the all tropical parts of the world, as far as I know. So please use your mosquito repellent when you're here, especially as it's the monsoon season. Okay, ApoBecs. First, let me say something about HIV. I am not going to talk about the life cycle, which is unbelievably complicated. But you may know that an important part of the HIV life cycle is an enzyme, a viral enzyme known as reverse transcriptase, which turns RNA into DNA. And it's a positive sense RNA virus. The virus particle carries the reverse transcriptase enzyme in the virus particle, and when it gets into a naive cell, the reverse transcriptase transcribes the single-stranded RNA using that as a template, and it makes an RNA-DNA heteroduplex. And then the ribonuclease H domain of the RT enzyme chews up the RNA strand of that duplex, leaving a single-stranded DNA, which is then used as a template by RT to make a second strand of DNA. So now we have double-stranded DNA. The double-stranded DNA gets into, this all happens in the cytoplasm. The double-stranded DNA goes into the nucleus of the cell and is integrated into the genome of the poor person who's been infected, and where it can sit for a very long period of time. If it wakes up and becomes active, then you have AIDS. And so that's a very stripped-down story about what happens to viral RNA in an HIV infection, but it's the essential piece for what I need to tell you today. So, of course, our immune system fights back. And one of the ways that our immune system fights back is with a group of proteins called ApoBec3, which are part of our innate immune system, not our adaptive one. And they act on single-stranded DNA. They are cytoplasmic proteins, and they act on single-stranded DNA. And these are proteins that have a variety of functions, but in this case, they are protecting our genomes from retroviruses and retroelements that would invade the genome. So what the ApoBec, there are many ApoBec proteins, but what the ApoBec3s do is they change a cytosine base into a uracil base by deaminating the cytosine. So if you do this on the single-stranded DNA, you change a whole bunch of C's into U's, then when you make the other strand where you were supposed to have had a G, you're going to have an A. So basically, you've changed the CG base pair into a TA base pair. And this, of course, effectively inactivates the virus. Of course, you won't be surprised to know that the virus fights back. This is, I should say, this catalytic activity is zinc dependent. The virus fights back, and the virus encodes another little protein called VIF. VIF stands for viral infectivity factor. And what the VIF protein does is recognize the ApoBec, binds to it, and recruits, which really means binds, also binds, another piece of protein machinery in the cell called an E3 ligase, which is an enzyme that polyubiquitinates the ApoBec3 protein, and that marks it for degradation by the proteasome. So basically, VIF puts ApoBec3s into the cellular trash can. And this whole little cycle is a molecular arms race. So we have, it turns out, an enormous number, we have a large number, enormous is an exaggeration, we have a large number of ApoBec proteins, and this little cartoon down here shows that the bottom is fish, and then we have cows and horses, cows and sheep here, these are mice. Each of these little arrows represents a different ApoBec3 protein, and here are primates. So there's been this expansion of proteins. These are all encoded in one locus in our genome. This is an expansion that's taken place to protect us from retroviruses and retroelements, all of this cytidine deaminase activity. So of the ApoBec, so ApoBec stands for apolipoprotein B RNA editing, mRNA editing enzyme, which is what some early discovered members of the family did. The ones I'm talking about don't act on RNA, they act on DNA, single-stranded DNA. And here's how they're organized. They have usually two homologous domains, sometimes one. They all bind zinc. There are several crystal structures and one or two NMR structures. Zinc is in here. It has 2 cysteine and 1 histidine ligand. The 4th position is available for the substrate. And there's a common catalytic glutamic acid side chain nearby. There are structures of these two domain, of the ApoBec3s, there is ApoBec3 or A3, A, B, C, D, F, G, and H in our genome. And F, G, and H restrict HIV. The others do other things. A3F and A3G have two of these domains in the mature protein. And the activities of these proteins are cytidine deaminase, RNA binding, and recognition by VIF. And so for the two domain, they call them Z domains, the two domain ApoBecs, catalysis is in one domain and RNA binding is in the other domain. And VIF binds wherever people have found it bound. Could be one or the other. The A3H protein has only one cytidine deaminase domain, which does all of these things. And there are no structures of that. There are no structures of any of them with bound RNA. And there are some, A3A, A3B, and A3C. There are a few crystal structures. Okay. These proteins are notoriously unstable, and so most of the ones that have structures known have been heavily mutagenized in order to get protein that can work, that you can work with in the lab and crystallize. Okay, so we collaborated on this project with Theodora Hatziano, who was a virologist at Rockefeller University, and she was working on developing a model for a primate model, a non-human primate model of AIDS in order to develop treatments, and was looking at the innate immune system and discovered that the A3H protein in the macaque was highly polymorphic. And she sent us, and these are 10 sites along the amino acid sequence, so this is about, they have between 180 and 210 amino acids. 10 sites where the sequence was variable among sequences that she obtained from 14 different animals. She sent us DNA for the, she examined their antiviral activity, so each of these is a different, this is a human one in the black data, and these are variants of the macaque ApoBec3H, and it's basically titrating in the plasmid that carries this into virus-infected cells and finding out how much antiviral activity there is. So they all have antiviral activity. When I say they're polymorphic, it doesn't mean that the A3H locus is duplicated. It means that there's mutagenesis, somatic mutagenesis taking place within the animals. And if you look for these proteins in the sequence database, there are many, many patient samples that have been sequenced and deposited. There are hundreds of sequences of A3H, human A3H, that have been obtained from patients. So it's actively trying to fight back against the virus. So we got some cDNA for these, and then my student, PhD student Jen Bone, got involved in the project and proceeded to purify A3H, and to our delight, it was actually a really nice protein to work with. No mutagenesis, it behaved itself, but it is tenaciously bound to RNA. So she treated protein with ribonuclease and got something on this overloaded radioactive gel, around about 10 nucleotides, and purified the human and then 3 variants and an inactive mutant of the macaque one, and extracted the nucleic acid, and you can see here it's purple, it's RNA. This shift in the band doesn't happen if you treat it with DNase, but after extensive ribonuclease treatment, this protein behaves on gel filtration like a dimer, apparently a dimer, and that peak is the ribonuclease. She tried lots of other things to get the nucleic acid off the protein, but did not succeed. And she also developed an assay and obtained catalytic activity, cytidine deaminase activity on a DNA, single-stranded DNA substrate, and with, a negative control with this catalytic dead mutant. Okay, so we have a bunch of variants, all have activity, and so then Jen grew some crystals, which presumably have RNA in them, based on, we now know, so this should have been solved by molecular replacement, because we know what these domains look like. The sequence identity is reasonably high. They all have the same fold, but it didn't work. Possibly she was new to crystallography when she did this, but there were 4 polypeptides in the asymmetric unit plus some RNA, and that might have been the reason, I don't know. But anyway, she decided to use the native zinc, and we estimate the anomalous signal, now that we know what the structure is about 2.8%. So it was easy to find the zincs, and the 4 copies helped to obtain a map, and she has a nice, about 2.25 angstrom structure. Okay, so what does it look like? Well, it looks like the rest of them. That's what the monomer looks like, the A3H polypeptide, but the big surprise came with the mode of RNA binding, which is like this. And this is not like any RNA protein complex that anybody has seen before, so that was kind of cool. And here is, so this was co-purifying RNA from an E. coli expression system. So this is no big deal, no special RNA, except for the fact that it's base paired. And you can see from this electron density, we have good density for the backbone, but the base pairs, all the base pairs have continuous density across the base pair, and they could be fit, each nucleotide could be equally well fit as a purine or a pyrimidine. There is no, it is a mixture of sequences. She, was hell-bent on proving that it really was RNA, so she deleted the 2' oxygen atoms and ran some refinement and made an omit map and, got density back, so we know it's RNA. Here's the structure of the RNA superimposed on an RNA duplex NMR structure, and then, so this is standard A-form duplex. Which is what RNA has, and it's also what RNA-DNA heteroduplex has. And so I found something in the PDB, and we superimposed that, and that's what it is. The interesting thing is how the protein interacts with the RNA. So as you might expect, there's no sequence similarity, no specific RNA sequence apart from base pairing. So there are no hydrogen bonds to bases. And all the contacts with bases in this complex are with aromatic side chains. So there are 9 nucleotides that we can see in the structure, which is nice since the gel tells us there's 10-ish in there. And nucleotides 3 through 9 are base paired. There's 7 base pairs you can see here. There's a tryptophan that stacks on the end and another one from the other subunit. And then there's this weird kink at the 5' end, and because there are very few base pairs, the kink brings the 2 5' bases next to each other, so they actually stack. And you can see them right here, and they're stacked between 2 tyrosine side chains, one from each polypeptide. So it's a very twofold symmetric structure. And then there's a little, nucleotide 2 is also not base paired, and there's a little aromatic pocket down here that base sticks into. This is just another view with some names of prominent and well-known loops in the ApoBec world. And there are a lot of charge interactions, basic amino acids with the nucleotide backbone. And the RNA is very, very well protected by the protein. About 40% of the nucleic acid surface is buried. And there are no contacts between the two protein molecules. They're totally mediated by the nucleic acid, so that if we could ever get the nucleic acid out of there, it would be a monomer. And you can see with these 90-degree away views, there is some mutagenesis that's been done that indicates that the VIF protein binds to these yellow amino acids, which are on 2 of the helices down here, well away from the RNA site and also the active site. So then we thought, well, is this the only one of these A3s that binds RNA like this? And it may well be. Here is the electrostatic surface looking down on the RNA duplex, and you can see how positively charged it is. This is the C-terminal helix of the protein, and there are many basic side chains that make salt bridges to the phosphates. And there is one other RNA binding domain from A3G, same orientation, looks like this in an electrostatic surface. You can see it's not very polar, and in fact there's a dimer contact. This is the C-terminal helix of A3G, so it does not look like that protein binds RNA in the same way. So then we went back to say, well, is this real? Is this some artifact? I mean, it looks very particular, the way the protein interacts, those lovely interactions with the RNA. So Paul Benash, who is a retrovirologist at Rockefeller, had previously taken A3H, A3F, and A3G. They get encapsulated into virus particles and then looked to see where on the genome they were bound, and did deep sequencing, cross-linked, and then deep sequencing to all the places that these proteins cross-linked. And so went back to those data. Here's a genome sequence, about 9,000 nucleotides, and then looked at the peaks, the place where there was most binding of A3H, and then the valleys, A through J. So the 10 highest peaks and the 10 deepest valleys where there was least binding, and said, okay, is it possible that there's any kind of a duplex formed in this region of the genome? Because the genome is very compressed when it gets packaged into virus particles. So as you're compressing the genome, maybe you're going to form a lot of stems. And in fact, there's a strong preference in the peaks for duplex, the ability to form duplexes, but not in the valleys. And then he looked at the data for A3G to say, well, where does it bind? And there is some duplex preference, but it's nothing like with H. So we think this is the only evidence right now we have that this is a real way that the protein binds RNA, but could be. And that brings me to the end of my story. And the acknowledgments for here are, first of all, this really gifted graduate student, Jen Bone, had help from Clay with the initial cloning for this project. And then Theodora Hatzianu and Paul Benash at Rockefeller. We have help from Alice Telesnitsky, a virologist at Michigan, and funding from various sources. And again, of course, the photons come from GMCA. So thank you very much for your attention, and I would be happy to take questions.
Thank you, and the floor is open for discussion. Please, your microphone.
Hello? Yeah. So I didn't quite catch where the short piece of RNA came from. So you pointed out positions on the HIV genome where it could originate from, but you would need a ribonuclease to cut it out, right?
So we made the protein in E. coli. Okay, for all of the enzymatic work and the structural biology, we made the protein in E. coli. Jen purified it in the usual way from E. coli, and I didn't show you the original gel filtration profiles before she treated it with ribonuclease, but it looks really ugly. And she ribonucleased it. So it is non-specific RNA from E. coli. It has nothing to do with HIV, and I don't think that there is, oh, they took it away. I don't think that there's anything particular about, I don't think there's any site specificity. This thing really wants to get into into virions, because it rides along with a reverse transcriptase. The A3 proteins are carried into new cells, and then when the single-stranded, when the DNA is made by RT, that's the substrate. And so the purpose of the protein is to kill the single-stranded DNA before it ever makes the second strand and gets integrated in the host genome. Does that make sense? So the only site that has, I think, functional significance is that people have also noticed that these proteins can restrict the virus even if you knock out the catalytic activity. So if you mutate that glutamate that's essential for catalysis, they can still inhibit reverse transcriptase. And the biggest, by far, the favorite place of A3H to bind is in the 5' UTR of the HIV genome, and there is an exactly 7 base pair. That part of the RNA, the untranslated region, is highly structured. And there's all kinds of contortions that it goes through that the HIV world is still trying to figure out during the virus life cycle. But there's a bit of 7 base pair worth of duplex in there that it turns out to be the highest hit place for A3H binding. And that may be significant because you need to, the RT needs to transcribe through there. And so in that way, that could be part of the special role of this protein. Oh, does that make sense, Udo? Okay.
Janet, that was very nice. I had a question about A3H protein again. Can you hear me clearly?
Yes, I can hear you. Yeah. Yes.
Okay.
Yes.
So it's very interesting that as I understood, the protein will bind to the RNA very tightly and also perform it will mutate the RNA.
Right. It has catalytic activity while bound to RNA.
Right.
That's correct.
So for it to then, I mean, at some point it would have to release the RNA so that the subsequent replication machinery is poisoned because the RNA has been mutated.
You know, it's not that neat and tidy. Yes and no. I think it binds wherever it can. It has different ways. I mean, I don't think we know how many copies get into a virus particle. I don't know, I don't think there's any evidence that it's processive. So our results that it has very nice catalytic activity with bound RNA is different than people thought. They thought that you had to get rid of the RNA in order to have it do the mutagenesis, and that's clearly not the case. It doesn't have to come, so here's what I think. Okay, this is I can't think of an experiment to show this cleanly, but when the DNA strand is made, okay, so I'm assuming that RT is going to be able to bump it off maybe at some point. Let's not worry about that. That's like an alternative. There's a couple different ways it can interfere. You have an RNA-DNA heteroduplex, and then the ribonuclease H domain is a remote domain of the reverse transcriptase. And the cartoon that we all have in our minds is that as this DNA strand is made, you have a heteroduplex, and then ribonuclease H is chewing off the RNA strand. Well, it turns out that the ribonuclease H is way, way slower than the transcriptase. And it's, somebody published a very nice study late last year of actually looking at the size of the bits that were left of RNA. You have a single strand of DNA and you have little bits of RNA left on there, and it turns out the average length is 6 of the little bits of RNA that are left on there. So I think one way that this protein could be working is to bind those little duplexy bits, which of course are A-form. And here you have protruding from both ends single-stranded DNA, ready to be, to be acted on by the A3H. I don't think the other A3 proteins work this way because it doesn't look like they have these same features, preference for binding to duplex.
Yeah.
Okay.
Yeah. No, it's quite remarkable. I mean, it seems that the protein has two options to sort of subvert the virus replication. One by causing mutagenesis in its genome, and possibly second by secluding RNA. But perhaps at some point the RNA has to come off the protein. So that's fine. I mean,
yes.
Yeah.
But there's a lot going on. Thanks, Amit.
Thanks.
Yes.
Hi. That was really interesting. I may have missed it, but are you able to find anything by mapping the polymorphic sequences on?
So the polymorphism, I was hoping could tell us where the VIF protein binds. So the VIF is very, the A3 proteins are extremely personal to the viruses that the animal that produces them has been exposed to. So if you take the monkey, the simian immunodeficiency virus, the HIV, the human virus, and you put it into a monkey, that monkey will not get sick because the A3 proteins in the monkey are very good because the HIV VIF does not recognize the macaque A3 proteins, and so they can't get degraded. They're not marked for degradation in the proteasome. If you take the if you take the simian immunodeficiency virus VIF sequence and put it into HIV, swap it out, and then put the SIV one in there, then you have a very good, then the macaque becomes actually a very good animal model for AIDS. The animals will get sick. It's really specific. And so we looked at where the variations are, where the differences between human A3H and macaque A3H, where they map on the structure, and that's going to tell us where VIF binds. Well, they're on the surface and they're kind of all over the place. There's one amino acid that somebody identified that seems to be key, and everybody else who's done mutagenesis started from that position and kind of worked in that neighborhood, so that's not very helpful. And the natural variants are there in other places, but, I think we're going to actually have to look at a VIF complex to really understand. And VIF is a ropey protein.
Thank you, Janet, for a beautiful presentation. And we have an appreciation moment coming.
On behalf of the organizers, it's my great pleasure to present a small token of appreciation to the speaker. Thank you very much.
Thank you. Thank you.
Also, it is my great pleasure to present a token of appreciation from the organizers to the chair also for organizing this event very successfully. Thank you very much.